Hello, and welcome to this tutorial describing the transgenic portion of the Center for Translational Environmental Health Research Targeted Genomics Facility Core.
The Targeted Genomics Facility Core provides access to powerful research technologies that will enable CTE-HR members to develop and test innovative hypotheses focusing on genome-environment interactions and their effects on human health. This core provides the opportunity to evaluate and validate new concepts in genetically relevant model systems as part of an integrated discovery pipeline involving hypothesis generation, testing, and translation. Use of this pipeline will accelerate the process of advancing innovative ideas and translating them for the improvement of environmental health and mitigation of environmental diseases.
Since September 2010, TIGM has resided in its own facility on the Texas A&M campus in College Station. The facility includes a molecular biology, cell biology, tissue culture, and surgery labs. The separation of a pathogen-free animal barrier facility and an open access vivarium allows development of clean mice while allowing mice from different sources to be brought to the facility, as well as collaborators to work in the open research core. TIGM also has a state-of-the-art cryogenic storage system that can be used by scientists.
Under contract, the TIGM C57 Black 6N gene trap library was made by Lexicon Genetics, now Lexicon Pharmaceuticals. Approximately 2/3 of the clones were made using a beta-geo vector and 1/3 with a neo-containing vector. 2 copies of the library were made. One copy of the library is housed in specially designed cryo rooms in both Houston and College Station. A total of 480,000 clones were produced, and sequence information was obtained for 350,000 of these. All of these clones are available to investigators.
External inquiries into TIGM's clones and cryopreserved mice are intuitive, and a search can be initiated using several criteria. If you cannot find a match in the database, don't be discouraged. Contact us and we will conduct a more thorough search through our database or provide alternative solutions.
TIGM provides center members with a variety of services. These include blastocyst injections with TIGM or external embryonic stem cells, embryonic stem cell manipulations, including electroporations, animal production, breeding, and maintenance, pronuclear injections, rederivations into pathogen-free environments via in vitro fertilization and embryo transfer, sperm and embryo cryopreservation, molecular biology services such as genotyping and vector construction, and certain animal studies.
TIGM employs several methods to produce genetically modified mice. Those include injecting DNA vectors and CRISPR-Cas9 DNA-RNA mixes into embryo pronuclei or targeted mutagenesis via ES injections into blastocysts.
DNA microinjection has traditionally been the main method to create transgenic animals. In general, only 5 to 10% of the treated eggs become transgenic progeny. Integration of a transgene needs to be confirmed with a sensitive genotyping assay, and it is important to confirm the expression of a transgene using RNA-based methods such as Northerns and RT-PCR, or using protein-based methods such as Westerns and ELISAs. The expression of a transgene may vary in transgenic offspring due to position effect and copy number, and in some cases a transgene may not pass through the germline. DNA microinjection has become very popular lately as an effective tool for CRISPR-Cas9-based genome editing.
Recently, TIGM adopted the CRISPR-Cas9-based genome editing tool, which includes a combined injection of DNA and RNA with the purpose of generating site-specific mutations in mice.
TIGM can effectively design and construct targeted vectors with LOXP elements positioned in the desired regions of genes and electroporate them into embryonic stem cells, which are then injected into blastocysts, resulting in generation of conditional knockouts, completely independent of clones existing in our library. TIGM can also procure mice expressing specific Cre recombinases, which can be bred into the mice carrying conditional mutations to generate tissue-specific or inducible knockouts.
TIGM can provide a variety of breeding services that can be used to obtain information about genetics, viability, and fertility of mouse lines. TIGM is serving as a centralized access hub to various phenotyping labs at Texas A&M University. For specific details and accommodations, please contact us and we will be glad to help you.
TIGM has developed a dedicated website detailing services, prices, and required paperwork to initiate transgenic services for Texas A&M University system faculty and for center faculty. The link to this portal is conveniently located on the center's website. Links to this portal are also found on the websites for Texas A&M University's Comparative Medicine Program, Health Science Center, and Institute for Genomic Medicine. If you cannot find the specific service you are interested in, please contact us and we will be glad to assist.